So what are the light reactions? We've moved to the light-dependent reactions, the part that requires light in order to occur directly. We're really talking about two major photosystem complexes and a couple of accessory proteins. All right. So the first photosystem is actually called Photosystem 2, and it's called Photosystem 2 because it was described or discovered second. The second photosystem is called Photosystem 1. You can eliminate all this issue by calling Photosystem 2 P680 and Photosystem 1 P700. P680 is the wavelength of light energy that the reaction center of Photosystem 2 absorbs light energy at. And P700 is the wavelength of light in nanometers, 700 nanometers, that the reaction center of Photosystem 1 absorbs light. All right. The photosystems are, just as the electron transport chain is in cellular respiration, is embedded in the membrane, although the membrane here is not going to be the inner mitochondrial membrane. The membrane here is called the thylakoid membrane. All right. Outside the thylakoid membrane is going to be the stroma, and inside is going to be the thylakoid space. All right. Now remember, we said that creating light energy, or creating energy from light, is a process that really is driven by the use of electrons, just as in cellular respiration. But the electrons here are not going to come from organic molecules, because that's what we're trying to make here. All right. The electrons are going to come from the splitting of water. All right. So generally, light energy enters through the leaves, through the cell, into the chloroplast, and basically gets received by the various pigments of what we call a photosystem, or a light harvesting complex, all right, which each of these photosystems has. Again, redox reactions are going to transfer electrons from a high energy state to a lower energy state. We're going to pump hydrogen ions across the membrane, all right, before the electron gets re-excited at a different energy level, P700, 700 nanometers this time, and to then get changed into another energy state where we have a reduction going on at the end here. But generally, the processes are the same, except the membranes which they're occurring, the organelles which they're occurring, and there's going to be a few major differences about where we get our electrons from and where electrons are going to go. All right. Specifically, the light harvesting complex is a complex all around here. It's embedded within this protein of many different molecules and atoms that are designed to transfer incoming light energy from one place to the next until it gets to the center, what we call the reaction center. We're going to play a couple videos here.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: Photosystem 2, cytochrome B6F, Photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and PI. which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. One photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem II are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem I complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
Pretty good there. All right, there's another video that we'll show as well. This one will also help you understand the process a little better.
System 2. The oxygen we breathe is a product of the photosystem II reaction. This animation will describe the processes that take place within this important complex. Photosystem II involves several key components, including photons, light-harvesting chlorophyll-binding proteins, a pair of chlorophyll molecules known as the P680 reaction center, phaeophytin molecules, and plastoquinones, along with water and oxygen. The reaction center of photosystem II consists of multiple proteins and pigment molecules. At the heart of the reaction center is a special pair of chlorophyll molecules, P680, which donate an electron to the electron transport system. In photosystem II, the electron is then passed to a phaeophytin molecule. The electron is then passed to plastoquinone QA and then to plastoquinone QB. These plastoquinone molecules are embedded in the D2 and D1 proteins. Once QB has accepted 2 electrons, it then acts as a mobile carrier to the next component of the photosynthetic electron transport system. Electrons from water are then transferred to the P680 molecules that have lost their electrons in the process. Additional proteins are involved in splitting the electrons from water. And still other proteins are necessary to build the complete photosystem reaction center. Surrounding the reaction center are light-harvesting chlorophyll-binding proteins. These proteins provide a way to harness the unique energy contained in light. When one of the many photons of light flooding a leaf hits a chlorophyll molecule surrounding the reaction center, it creates resonance energy. You can think of this as vibrational energy. Here you can see the chlorophyll vibrating. That resonance or vibrational energy is then passed to a neighboring chlorophyll molecule. It is then passed through several chlorophyll molecules until it reaches the P680 reaction center. It is that energy that results in the loss of an electron from the P680 molecule. Now we can see how these processes work as a whole. First, a photon of light activates a chlorophyll molecule. The resonance, or vibrational energy, is transferred to the P680 molecules. An electron is lost from P680. It is then donated to QA, then to QB. The P680 molecules are then reduced by the addition of an electron generated by the splitting of water molecules at the oxygen-evolving complex. Since QB needs 2 electrons to become mobile, a second photon of light is required. The resonance energy is again transferred to the reaction center. An electron is lost from P680 and transferred via QA to the QB, which already contains 1 electron. The fully reduced QB is then transferred to the cytochrome B6F complex. The P680 molecules are again reduced by the oxygen-evolving complex. Here you can see water being split at the oxygen-evolving complex. 2 water molecules must be split to provide electrons to reduce P680. The oxygen we breathe is a product of this water splitting process.
All right, back to it. So those are the the processes of the electron transport generally. Well, we're going to talk a little bit more about that. The important thing to know, the photosystem is the entire complex. All right. Now the photosystem includes the light harvesting reactions, all right, the light harvesting complex, and it includes the reaction center, the photosystem. Okay. Now the reaction center is a pair of chlorophyll A molecules. All right. So all of the pigments that surround a photosystem are really a mix of chlorophyll A and B and carotenoids and different types of accessory pigments that are designed to take in as much light energy as possible, absorb as many photons at different wavelengths. This energy gets resonated and transferred and absorbed around the photosystem to different pigments until it reaches the reaction center chlorophyll molecules. All right, these 2 reaction center chlorophyll molecules lose their electrons to a part of the redox reactions that go on in the photosystem, and then that energy splits water and takes electrons one by one from the hydrogens of the water molecule. All right. Now, photosystem 2 has a reaction center called P680 because that's where it absorbs the most light at, raising its energy state. And photosystem 1 is also called P700 because that's the energy level that it works at. Okay. I actually don't know why they work at such different levels. All right. But it just makes sense that the electrons in photosystem 2, by the time they get there, are likely to be less energized because their energy has been used to do work, and it takes a lower wavelength or a longer wavelength, lower energy wavelength in order to excite them to another level again. All right, that'd be something to research on your own if you're really interested on why one has a reaction center at 680 nanometers and why another one has a reaction center at 700 nanometers wavelength. All right, this is just a more complex and a little more descriptive diagram of all the redox reactions that are going on at one time. All right. The redox reactions, again, are designed to transfer electrons from one place to another so that we can use these electrons, number one, to reduce NADP plus to NADPH, and also to use these electrons to pump hydrogens across the membrane. All right. Again, we're using the electrons to pump and release hydrogens across the membrane, all right, into the thylakoid space, all right, by which ATP synthase will then use this proton gradient to make ATP. Right? So now what we end up with is lots of ATP from ATP synthase and lots of reduced NADPH to carry electrons to the Calvin cycle, or the light-independent reactions. All right. So some important things to know is the difference between the reaction center and a light harvesting complex, also called antenna pigments. All right. And where the electrons are coming from, where is things happening? All right. What is the point of our mobile carriers? All right. What is the point of our plastoquinocytochrome B6 complex? What is the point of this complex here? All right. And a little bit about how these two photosystems differ from one another. Photosystem 1, P700, is actually the photosystem that reduces NADP plus to NADPH. So it's really the one that provides us with our mobile electron carrier so that we can have reductive power later on. All right. But all of this stuff is being energized by photons coming from light, which is really important. All right. There's just another example of how extremely complex all these proteins are. This is photosystem 2. This is the cytochrome complex, and you can see this is a channel to allow hydrogen ions to come through using the energy from electrons. We also allow hydrogen ions to come through as the mobile carriers move across from one place to another. And also we have our photosystem 1 here with our ferredoxin. Ferredoxin is basically the molecule that is going to be reduced, and it's going to help us reduce NADP plus to NADPH using those electrons taken from initially what would be water. Remember, in cellular respiration, ATP synthase is taking an electron gradient that is higher in the mitochondrial inner mitochondrial membrane, all right, in the inner membrane space, I mean, and the electrons are flowing into the mitochondrial matrix. On the other hand, in photosynthesis, your hydrogen ion gradient is being created in the thylakoid space, and the hydrogen ions are being allowed to flow through the ATP synthase into the stroma. And that's important because ATP, and NADPH are going to be used for the Calvin cycle, a reductive process that also occurs in the stroma. All right. So again, light energy energizes pigments that transfer energy to the reaction center where electrons are being taken from the splitting of water. Hydrogen ions and oxygen are created. All right. We have mobile carriers here, and we have cytochrome B here, which is also going to help pump hydrogens across the membrane. And we have our photosystem 1 here, which again takes the electron. It doesn't split water because we already have the electrons, but light energy is transferred from the light harvesting complex to the reaction center where electrons are transferred via redox reactions to ferredoxin, where we can make NADPH from NADP+, and we can then make ATP from the hydrogen ion gradient and ATP synthase. All right. So this process doesn't differ much, except where it happens on the membrane it happens, where we get our electrons, where our energy is going to come from. So it would be helpful to focus on the differences between the two. Just another perspective here, the light reactions, these reactions here are occurring on the thylakoid membrane. We are going to be pumping hydrogen ions into the thylakoid space, allowing them to go into the stroma. The important parts of the light reactions are to create ATP and to reduce NADP plus to NADPH using electrons, light energy, water, creating oxygen as a byproduct. Now there's one more thing that can happen in many organisms. It's called cyclic electron flow. All right. Remember we talked about feedback and the things that happen if the cell realizes that it doesn't actually need something. Well, let's say that the cell doesn't need to make any more sugar. All right. For some reason, it's not growing. It doesn't need to make cellulose. It's not able to make starch. Or for some reason, it just doesn't need for the Calvin cycle to occur. The Calvin cycle's point or purpose is to create sugars, right? Well, let's say the cell doesn't need to create sugars. Maybe NADPH is building up. If NADPH builds up, then the electrons are not going to be transferred onto ferredoxin. All right. Well, actually, yes, the electrons will be transferred onto ferredoxin, but ferredoxin, instead of taking the electrons to NADP+, it will take the electrons back through the cytochrome B6 complex, whereby the electrons will then go back to plastiquinone and release them again to photosystem 1. So here, we're taking electrons and we're cycling the electrons through, all right, which means that we can create that hydrogen ion gradient necessary to create ATP, all right, and we can also reuse our electrons, and all we need is light energy to continuously excite the accessory pigments and P700, or the reaction center. All right. So cyclic electron flow is a way for organisms to actually react to not needing sugars, but still needing ATP. All right. Why would an organism make sugar and then break it down for energy if it could just make energy right away? So cyclic electron flow is a way to make ATP directly and not have to go through the process of the Calvin cycle in order to make sugars and then send those sugars to the mitochondria where then energy can be made. So it's we're saving a bunch of steps by being able to use cyclic electron flow. It is also thought that photosystem 1 was actually the first photosystem to actually exist because it's found in many very, very simple organisms, and that cyclic electron flow might have been the only method of making energy for many very simple organisms in evolutionary history. So overall, what's going on here? We have the organelle called the chloroplast in the plant cell. It's a double membrane cell. The chloroplast houses these things called thylakoids, which a stack of them are called grana. Many stacks are called grana. These thylakoids have embedded on them photosystem 1 and photosystem 2 and some other membrane proteins and mobile carriers that are designed to make ATP and NADPH from NADP plus and NADP using electrons from water, all right, creating oxygen as a byproduct, and using the energy from light in order to excite the photosystems to break water and move electrons, create a hydrogen ion gradient, and then make ATP from ATP synthase from the hydrogen ion gradient, and reduce NADPH using the electrons that we took from water originally that came from the chlorophyll A molecules. Wow, to be a rock star, Def Leppard, man, what it would have been like to live in the 80s, or to actually be an 80s rock star. I actually did live in the 80s. I always forget that sometimes. But couldn't have been as cool as Def Leppard, and definitely didn't write a cool song like Pour Some Sugar on Me. But you can imagine why the song is relative. Because the plant cell is going to be pouring some sugar on itself. Yes, the plant cell is going to be making its own sugar. So it's more like, make some of my own sugar. Yeah, 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 make some of my own sugar. So The Calvin cycle is the second part of the light reaction. It says, whoa, 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 back up. It's the second part of photosynthesis that involves taking carbon dioxide and actually fixing it or reducing it and building it from carbon dioxide to organic molecules, phosphoglycerate, bisphosphoglycerate, glyceraldehyde 3-phosphate, also known as G3P, and then making a G3P sugar from it. All right. So some of the important things about the Calvin cycle, there's three main phases of it. It's carbon fixation. Fixation is basically taking RuBisCo, which is an enzyme, is going to take 3 carbon dioxides, all right, and join it with 3 Ribulose bisphosphates. Important thing to do is count carbons. We have 3 molecules, 3 5-carbon molecules. That's 18 carbons. All right. Sorry, 15 carbons. 3 carbon dioxides go in, and now we have 18 carbons. Now what happens here is in the carbon fixation process, RuBisCo, which is an enzyme, binds to carbon dioxides, multiple number of them, and actually puts the carbons onto already existing ribulose bisphosphates, also known as RuBP. This spontaneously leads to these 3 carbon molecules dissociating into 6 3-carbon phosphoglycerates. All right. Now remember where we've seen phosphoglycerate. All right. We saw phosphoglycerate in glycolysis. Remember, glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate, fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Dihydroxyacetone phosphate goes to glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate goes to 1,3-bisphosphoglycerate, and from 1,3-bisphosphoglycerate it goes to 3-phosphoglycerate, and then from 3-phosphoglycerate to 2-phosphoglycerate, and then from 2-phosphoglycerate to phosphoenolpyruvate, and then phosphoenolpyruvate to pyruvate. So it's interesting that the Calvin cycle does something very, very similar, except notice here it's going in the other direction. So in glycolysis, we go from 1,3-bisphosphoglycerate to 3-phosphoglycerate. In glycolysis. But in the Calvin cycle, we're now starting with 6 3-phosphoglycerate molecules, all right, which 18 carbons. We're using energy from ATP, all right, to phosphorylate these 6 molecules using 6 ATP. Higher its energy state, now we have 1,3-bisphosphoglycerate. We further add hydrogens and electrons to this molecule to make it even more high energy, and now we have 6 G3P, or 6 glyceraldehyde 3-phosphates. Remember, the Calvin cycle, this whole thing is occurring in the stroma, getting its energy from things that were made in the light reactions. One of the glyceraldehyde 3-phosphate molecules is going to leave to either be used directly, probably not, all right, or to be made into a more complex sugar like starch or cellulose, or in many cases sucrose, because that's how sugars are usually transported in plants. All right. So now we get one glyceraldehyde 3-phosphate molecule, and the rest of them will then get phosphorylated again and made into 3 5-carbon molecules again. These 3 5-carbon molecules again will come on around. RuBisCo will bind to 3 more carbon dioxides. Carbon dioxides will bond, spontaneous splitting into 6 3-carbon molecules. We add more ATP, more NADPH. More reduction. We'll lose another glyceraldehyde phosphate, and that's basically how it happens. All right. So the important thing here is that RuBisCo can actually bind to carbon dioxide, and then take carbon dioxide and put it into a form that can be used in organic molecules. All right. So this is really the most important process about the Calvin cycle, is the ability for an enzyme, RuBisCo, to bind to carbon dioxide and actually put it into organic form, something that no other organisms are able to do. Take carbon dioxide out of the air and put it into an organic form. This is basically how carbon gets fixed and sequestered from the atmosphere into living organisms. This is the same carbon that humans and animals and mammals and all other organisms are going to use to build themselves up. This is the same carbon. It came from carbon dioxide. All right? The Calvin cycle, beautiful process. 3 phases: carbon fixation, taking carbon, putting it into organic form from carbon dioxide, reduction using ATP and NADPH, and regeneration using more ATP. So generally, we use 9 ATP and we use 6 NADPH in order for one cycle to occur, just to make one glyceraldehyde 3-phosphate. All right, there's a lot going on here. Now, RuBisCo is extremely important. It's able to take carbon dioxide and fix it or bind to it and then put it onto Ribulose bisphosphate. All right. Now, the crazy thing about RuBisCo, it's actually the most common enzyme in the world. It's called Ribulose bisphosphate coenzyme. Or no, sorry. Ribulose bisphosphate carboxylase oxygenase. Whoa, what a name. It's called a carboxylase because it binds to carbon dioxide. And it's called oxygenase because it actually has the ability also to bind to oxygen. Crazy thing is that 8 active sites. This enzyme is so complex, there's 8 active sites. 8 places for carbon dioxide to bind and to attach to RuBP. So there's 8 sites where CO2 can be fixed onto the RuBP. Now, one of the crazy things about RuBisCo is it has the tendency to bind also to oxygen. All right. Now, I want you to think about why this is not a good idea. Or not necessarily a good idea, but why is this not beneficial for the plants? All right. I want to talk about this more in class. But why is it bad that RuBisCo also has the ability to bind to oxygen as well as carbon dioxide? All right. What is the significance of that? I also want you to try to think about the question of this. All right. How, evolutionarily speaking, is it significant that RuBisCo, designed to fix carbon dioxide into organic molecules, how could it have even been synthesized to bind to oxygen as well as carbon dioxide? So a couple questions to answer. Why is it important negatively that RuBisCo can bind to oxygen? And how evolutionarily do you think this came about or became of some importance? All right. It's a great question and something I love to discuss. So I'm not going to go over it totally now, but please ask me in class because it's very important to know. All right. So RuBisCo binds to carbon dioxide and oxygen, although carbon dioxide is the aspect of it that fixes it into an organic molecule. So, back to the big picture. All right. Water plus CO2 plus light energy yields oxygen and sugar. Okay? More specifically, water gets split for its electrons using light energy to make a hydrogen ion gradient to make ATP, and the electrons are used to reduce NADPH. ATP will phosphorylate molecules in the Calvin cycle. NADPH will reduce molecules in the Calvin cycle. We create one glyceraldehyde 3-phosphate per turn of the Calvin cycle. This occurs in the stroma. The light reactions occur on the thylakoid membrane, in the thylakoid space, and a little bit in the stroma. All right, but all photosynthesis occurs in a chloroplast cell. Crazy, 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 crazy. All right. Now, a couple really cool adaptations. One of them called C4 photosynthesis, and one called crassulacean acid metabolism. But basically, one of them, all right, in C4 photosynthesis, carbon dioxide is bound to an organic molecule called phosphoenolpyruvate, which you've heard before as well. All right, is sequestered into a 4-carbon compound and then released into another cell called the bundle sheath cell, whereby carbon dioxide can bind to RuBisCo. Now, this adaptation, C4 photosynthesis, is directly related to the binding of RuBisCo with carbon dioxide and oxygen, and actually solves the problem of RuBisCo binding to both, because it is a problem. All right. And this is called C4 photosynthesis. The important thing is that normal photosynthesis is called C3 photosynthesis because the first molecule in carbon fixation is basically a 3-carbon molecule. You have 6 of them. That's why it's called C3 photosynthesis. The adaptation, C4 photosynthesis, sequesters carbon dioxide in an organic compound to be released in a bundle sheath cell where only RuBisCo can bind it. Now, crassulacean acid metabolism is a type of photosynthesis where carbon dioxide is actually sequestered as an organic acid, as oxalic acid and malate, so that it can be released in the daytime in chloroplast cells. Now, CAM photosynthesis is an adaptation to living in extremely hot and arid conditions. The plant cannot be opening its stomata during the day because it's losing too much water vapor. So actually, it is designed to pick up carbon dioxide at night, sequester it, and release it during the day so that the Calvin cycle can run and the plant doesn't have to open its stomata to collect carbon dioxide during the daytime. All right? So these are two major adaptations to photosynthesis that occur in plants generally in very hot and arid conditions like the desert for CAM photosynthesis, and organisms that usually occur in the tropics, those that can use more sunlight or actually are competing with other organisms year-round, especially in the tropics. These organisms tend to be C4 photosynthesizers. All right. So some important things to know: the parts of a plant cell, where things happen, and definitely comparison of cellular respiration with photosynthesis. I am going to, well, I think we can go probably a little bit further here. We've got a couple more minutes. Important things to know is that carbon dioxide is what we call a greenhouse gas. All right, it is a gas that as sunlight enters the earth, it actually keeps, it does a couple things. Number one, it prevents sunlight from entering the Earth's atmosphere by reflecting it and absorbing that energy. But also what it does is it keeps the Earth warm. All right, so carbon dioxide is really what prevents a lot of the Earth's warm air from escaping by absorbing that heat and readmitting it back to the Earth's surface. All right. Now you can imagine that with so much carbon dioxide being released into the atmosphere by fossil fuels, because remember, fossil fuels are really old plant material that was liquefied under high heat and high pressure. All right. It's basically just carbon dioxide. All right. When you burn gasoline, when you burn things in a fire, all right, when you burn things for electricity like coal and oil and natural gas, all you're doing is releasing that carbon dioxide into the atmosphere in massive amounts. All right, and what this happens is it increases the effect of the greenhouse, and it basically keeps warm air trapped in because of the higher levels of carbon dioxide in the air, and leads to a warmer planet. Now, if you look here, researchers know this because you can measure the dissolved oxygen and carbon dioxide bubbles that occur in the microenvironments in the Antarctic and the Arctic. The ice sheets actually collect a certain amount of carbon dioxide in the air and sequester it within the ice. All right. So what scientists are able to do is measure the levels of carbon dioxide in the air long periods of time ago, periods at which we weren't even, civilized organisms. This is like 200,000 years ago, and compare it to the carbon dioxide levels of today. And as you can see, the carbon dioxide levels are so high today that they are obviously having an effect on the Earth. The fact that the greenhouse gases are having an effect on the Earth and warming the planet actually is not a point of contention. I repeat, it's not a point of contention. It's not even my opinion. We're burning so many fossil fuels, releasing so much carbon dioxide into the air. Plants are not taking it up because plant growth is not nearly as fast as we are releasing carbon dioxide emissions in the air. Just imagine how much gasoline is burned a day. And the lights that are probably going on in your house right now, those lights are being powered not by alternative energy, they're being powered by coal and fossil fuel plants somewhere in your area where you can't even see that happening. So right now, if there's a light on, and this computer is actually being run by fossil fuels. So crazy. All right. Here's another example of different greenhouse gases. Carbon dioxide, methane, and nitrous oxide are all greenhouse gases. They all help in reflecting light energy and by reabsorbing heat that is lost from reflection on the Earth's surface. All right. As you can see, the amount of nitrous oxide, methane, carbon dioxide has gone up exponentially in a very, very short period of time. This is only 2000. This was taken. This map was done about 10 years ago, 7 years ago. So you can imagine where this trend is now, because we're not burning less, by the way. We're not releasing less carbon dioxide. And now we're looking at global temperatures from 1880 to 2010. All right. As you can see here, the global temperature is on the rise. All right. It is of no coincidence that carbon dioxide levels and rising global temperatures are happening at the same time. We know carbon dioxide is a greenhouse gas. We know that global temperatures are rising. It's not actually being disputed about whether or not the global climate is changing, because it is. And it's also not a point of contention about whether or not there have been past climate changes on the Earth. There have. One of the important things is, number one, it's being caused by human activity, which is different. Number two, the scale at which it is occurring at unprecedented levels. All right, and this all is important to our daily lives. You know, I lived in an era in which acid rain and global warming were seen as not big of a deal. Acid rain was proven to be a huge deal to organisms in lakes and rivers of the northeastern United States. Acid rain has since actually been controlled. Global warming, on the other hand, is something that is the whole world is trying to wrap itself around. And in all honesty, it is going to be up to you, for those of you that are kind of be going into fields that are going to be solving problems of the future, to figure out ways not just to decrease the carbon footprint that we have, right? The carbon footprint is the amount of carbon that our activities release into the atmosphere. But it's also going to be to what are some ways in which we can change our day-to-day activities and methods of energy consumption so that our footprint is much lower. All right. So thank you for listening. I know it was a lot of stuff. I also know that we're getting close to exams, but I'm going to make sure that you guys are prepared, that you have all the things that you need to be prepared as far as study guides and ample review time. Thank you for getting through the lecture, and please stay open and write down any questions that you have so we can answer them in class. All right? The more dialogue that we have going in class, the better we can together take on these concepts and master them as one. So I look forward to talking about these things in class, and I also look forward to your questions. Thanks. Have a good night or day or evening or morning.